Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore the urea cycle. Imagine your body is a bustling city, constantly building and tearing down structures. This process generates dangerous waste that must be handled carefully. We will discover exactly how your liver acts as a highly advanced waste treatment plant to keep you alive and healthy. To understand why the urea cycle matters, we first need to look at protein metabolism. Every single day, your body breaks down old proteins from muscles and enzymes. When these cellular buildings are torn down, they leave behind toxic rubble in the form of ammonia. This is essentially the same harsh chemical you might use to clean your house. If this free ammonia is allowed to build up in your bloodstream, it acts as a severe poison, particularly to your delicate brain cells. Therefore, the body desperately needs a way to neutralize it. Now that we know how dangerous ammonia is, let us see how the body handles it. To prevent toxicity, your liver steps in as a highly advanced waste treatment plant. It takes that dangerous ammonia and carefully packages it into a harmless, water-soluble compound called urea, which your kidneys can safely flush away. This packaging process is called the urea cycle, or the ornithine cycle, discovered in 1932. It involves 5 precise biochemical reactions. The first 2 happen deep inside the mitochondria, and the remaining 3 occur in the cytosol. By constantly regenerating its starting material, ornithine, the cycle runs endlessly. Understanding this cycle is not just academic. It is a matter of life and death. Without the urea cycle, human life would be impossible. When this cycle fails, either due to genetic defects in newborns or severe liver disease in adults, ammonia rapidly accumulates in the blood. This triggers a life-threatening condition known as hyperammonemia. The toxic buildup leads to severe brain swelling, confusion, seizures, and eventually a fatal coma if left untreated. This is exactly why doctors routinely monitor blood urea nitrogen levels in patients, as it tells them how well the liver and kidneys are managing this critical waste disposal. This brings us to the overall chemical equation that summarizes the entire urea cycle. Conceptually, this formula shows us all the raw materials going in and the final products coming out. Let us look at the symbols. NH3 represents the toxic free ammonia. HCO3- is bicarbonate, providing the carbon atom. C4H7NO4 is aspartate, donating the second nitrogen. On the product side, CH4N2O is our safe, water-soluble urea. Notice the energy molecules. 3 molecules of ATP are consumed to drive this process forward, breaking down into ADP, AMP, and inorganic phosphates. If ammonia levels increase, this cycle must run faster, consuming significantly more cellular energy to keep you safe. Before we move on, let us see this with a concrete example regarding energy expenditure. A student is asked to determine the energy cost to synthesize 1 molecule of urea. We are given the overall stoichiometric equation we just looked at. First, we identify the ATP molecules on the reactant side. The equation clearly shows that exactly 3 molecules of ATP are consumed. So our initial answer is that 3 ATP molecules are used. However, simply counting the molecules does not tell us the true metabolic tax the body pays. We need to look deeper at the high-energy phosphate bonds. So we have established that 3 ATP molecules are used. This brings us to deriving the true energy cost by counting the actual high-energy phosphate bonds broken. We will split this into 2 parts. In the very first step of the cycle, The enzyme carbamoyl phosphate synthetase 1 consumes 2 molecules of ATP. As you can see in the reaction, each ATP is converted into ADP. Mathematically, converting one ATP to ADP breaks exactly one high-energy phosphate bond. Since 2 molecules are used, this initial gatekeeping step costs the cell exactly 2 high-energy bonds to activate the ammonia. Now, let us continue our derivation to find the final result. Later in the cycle, the enzyme argininosuccinate synthetase consumes the third and final molecule of ATP. However, the mathematical reasoning here is different. This time, the ATP is cleaved into AMP and pyrophosphate. Cleaving ATP all the way down to AMP breaks both of its high-energy phosphate bonds simultaneously. Adding these together, we have 2 bonds broken in the first step and 2 bonds broken here. This gives a total of 4 high-energy phosphate bonds consumed. This proves that safely disposing of nitrogen is a highly energy-expensive process. Now that we understand the energy cost, let us look at how this applies in a hospital setting. When a patient consumes a high-protein diet, their body generates excess nitrogen that must be excreted. Clinical dietitians must calculate exactly how much urea the liver needs to synthesize to safely clear this nitrogen. This is especially critical when managing patients with severe kidney or liver failure to ensure toxins do not accumulate. To perform this clinical calculation, we first need to understand the chemical makeup of urea, specifically its molar mass and the fraction of that mass that comes from nitrogen. This brings us to the formula for the molar mass of urea. Conceptually, molar mass tells us how heavy 1 mole of a substance is. The chemical formula for urea is CH4N2O. To find the total molar mass, represented by the variable m, we sum the atomic weights of each element. Carbon is 12, hydrogen is 1, nitrogen is 14, and oxygen is 16. Notice that the formula contains exactly 2 nitrogen atoms. This pair of nitrogen atoms is the key to urea's efficiency as a waste carrier. If it only carried 1, the body would spend twice as much energy. Before we calculate the patient's clearance, let us see this with a concrete example of finding the nitrogen mass. We are asked to find how much of urea's 60 grams comes strictly from nitrogen. We define the variable m sub n as the total mass of nitrogen in 1 mole. Since we know there are 2 nitrogen atoms in every molecule, we multiply the atomic weight of nitrogen, which is 14, by 2. This gives us 28. Therefore, out of the total 60 grams of a mole of urea, exactly 28 grams are pure nitrogen waste. Now that we have our molar masses, we can introduce the nitrogen clearance proportion formula. Conceptually, this formula uses a simple ratio to scale up the amount of waste to the amount of packaging needed. The variable mu represents the final mass of urea the liver must synthesize. We take the given excess nitrogen, represented by Me, and multiply it by the ratio of urea's total mass, which is 60, divided by its nitrogen mass, which is 28. If the patient's excess nitrogen increases, the required mass of urea will increase proportionally, demanding more work from the liver. Let us apply this formula to our clinical example. We are given that a patient generates 1.4 grams of excess nitrogen per day. We need to find out how many grams of urea the liver must synthesize. We set up our proportion, plugging in 1.4 for the excess nitrogen, and multiply it by the fraction 60 over 28. The math simplifies beautifully, and we find that the liver must synthesize exactly 3.0 grams of urea. This physically demonstrates how efficiently the liver packages waste, using a small amount of urea to carry away significant toxicity. Now that we have used the formulas and calculations, let us look at the complete picture. Here, you are seeing the full 5-step urea cycle. Notice how the diagram is split into two distinct cellular compartments. The top blue region represents the mitochondrial matrix, where the cycle begins. Here, ammonia and bicarbonate are fused into carbamoyl phosphate, which then joins with ornithine to form citrulline. Follow the arrow as citrulline crosses the double membrane into the yellow cytosol region. Here, 3 more enzymes act in sequence, eventually releasing the harmless urea molecule at the bottom right and regenerating ornithine to keep the cycle turning. We have covered a lot of ground today, so let us summarize the key takeaways. First, the urea cycle is an essential metabolic pathway that protects your brain by converting toxic ammonia into harmless urea. Second, this elegant cycle bridges the mitochondria and the cytosol, running continuously by regenerating ornithine. Third, we proved that this safety comes at a high metabolic cost, requiring 4 high-energy phosphate bonds for every molecule of urea produced. Finally, understanding these chemical proportions allows doctors to calculate exact nitrogen clearance, saving lives in the clinic. You now have a solid understanding of the urea cycle.